It's great to introduce Dan Ha, who is a postdoc in my lab. He has been in the lab for about 3 years, I think. Yeah, he has been working on many different projects involving multiomics integration to understand different biological questions. And he's also a QCBio postdoc fellow, so has been supporting a lot of projects, data mining projects across the campus as well. Very productive and amazing scientist. So yeah, that's my introduction for Dan. I hope you enjoy his presentation.
All right, thank you, Shia, for really nice introduction. Can everyone see my screen?
Yeah.
Yes.
Okay. Thank you. Okay, so hi everyone. So for today's talk, I'm going to talk about the anti-obesity effect of Akkermansia muciniphila, specifically under fructose condition, using the multiomics, 3 different multiomics technology. So a little bit of introduction of Akkermansia muciniphila. It is considered one of the next generation beneficiary bacteria, where it is known to inversely associated with metabolic syndrome and diseases such as obesity, diabetes, and inflammation. As the name indicates, the mucin and phila, the bacteria itself loves the mucin in our gut. So in our intestinal epithelial cell, there is an extra layer of barrier that covers the intestinal cells, which is known as the mucous layer. Usually, is generally accepted that the thicker the mucous layer, the more healthier you are, because it provides extra protection against potentially pathogenic bacteria in the gut. So even though the Akkermansia degrades the mucins in the gut, there are so many beneficiary effects, and as a result, it actually helps the gut epithelial cells to produce more mucins. So as a result, the thickness actually get much thicker with the Akkermansia treatment, which was demonstrated by Van der Lut et al. Study in 2019. And a lot of beneficiary mechanisms have been already discovered with the Akkermansia muciniphila, specifically, for example, the short-chain fatty acid here. Akkermansia synthesized. Akkermansia actually involved in the production of short-chain fatty acid. Which causes an extra benefit, and it also involved in modulation of gut permeability, which inhibits the entrance of the endotoxins. It is involved in lipid metabolism and immune functions. It also involved in the conversion, inhibits the conversion of TMA into TMAO, which is one of the key toxic molecule involved in liver disease, progression of liver disease such as NAFLD, non-alcoholic fatty acid liver disease. But despite these known mechanisms, a lot of the studies were conducted under high-fat diet. So these are the results from the primitive PubMed search. So I searched for Akkermansia with the high fat, and the result indicates that there were like 172 studies. Where if I search for high fructose, there was only like 8 study there. But understanding of fructose metabolism is important, especially now with the modern diet, because we as a human, we rarely consumed fructose in high amount before the mass production. So overdose of fructose can cause impairment of blood lipids, NAFLD, and as well as insulin resistance, which in turn resulted in obesity and type 2 diabetes. And it is also important to note that fructose does not suppress the appetite as much as the glucose does. So overconsumption of fructose is quite common. In the previous study, our lab conducted actually fructose-related mouse research. So both this research actually used three different mouse strains and see their response to the high fructose diet. The first study actually looked the transcriptomic change using the RNA-seq. The second study actually looked at the microbiome change with respect to the fructose consumption. So long story short, we had shown that out of three different strains of mice, only DBA mice showed the obesity state with the high fructose consumption, while other two bacteria really didn't have much of a obesity phenotypes, even though they were consumed with the high fructose. And turns out that these two mice strains contains higher proportion of Akkermansia in them, which is quite negatively correlated with the adiposity gain sign here. So the black dot here represents the DBA mice strain, which obviously you can see that Akkermansia proportion is quite low compared to the other strains, and their adiposity gain was quite increased. And this box plot also shows that DBA mice contain a lower level of Akkermansia compared to the other strains. We also did some correlation analysis between transcriptomic data and Akkermansia in this study, and we also discovered that their hypothalamus showed the highest number of genes that are highly correlated with Akkermansia proportion. And hypothalamus is quite important when it comes to maintaining our body's homeostasis. It's a central power in the unit. It is literally in the center region in our brain, and it controls the body temperature, thirst, appetite, and weight control, as well as emotions, sleep cycles, and blood pressure. I also want to emphasize that the gut and brain actually communicates each other through the gut-brain axis, through the vagus nerve. So whatever you have in the gut can exert either a beneficiary or pathogenic effect to the brain through the gut-brain axis. So having this as a background knowledge, we decided to dig a little deeper into how Akkermansia actually exerts this beneficiary effects. So we did three different omics data analysis here, microbiome, using the 16S rRNA, and metabolomics from fecal samples and plasma samples from the mice. And we also did hypothalamus tissue single-cell RNA-seq. And this is the overall study design. It's total of 8-week experiment. So we so DBA mice was used for this experiment. So we treated the mice with antibiotic treatment to wipe out the gut microbiome initially, and then we started the administration of Akkermansia with two different diet conditions, fructose and water. So as a result, we have the four groups: Akkermansia with fructose, control with fructose, Akkermansia with water, and control plus water. So in terms of the phenotype, we, as expected, we did see the Akkermansia actually inhibited some of the obesity phenotypes. So here, DBA mice with Akkermansia and fructose had the lowest body weight gain compared to the control group, which is the black dot here. Total calorie intake, there wasn't any statistical significance there, but despite this, we did see some significant increase in fat mass in the control group, which is somewhat expected. But in the Akkermansia treated group, we didn't see the the significant change in the fat mass gain, which also confirms that the Akkermansia exerted some anti-obesity effects. In the microbiome level, so this first plot here represents the PCOA analysis using the beta diversity indexes from each sample. It nicely separated the samples, the control samples and Akkermansia treated samples. And the Firmicute bacteroidetes ratio, also known as the FB ratio, this ratio is an indicative value for obesity state. So usually the high FB ratio indicates the obesity state. That's what we observed in the control group. We see the high FB ratio and the significantly decreased FB ratio in the Akkermansia treated group. In the individual genus level data, we obviously see significant enrichment of Akkermansia genus because in the Akkermansia treated samples. Interestingly, we also observed that lactobacillus, which is also one of the beneficiary bacteria, was significantly enriched with Akkermansia treatment. Conversely, some potentially pathogenic bacteria such as Staphylococcus and Aerococcus was somewhat downregulated upon the treatment of Akkermansia. Oh, sorry. So in the metabolomics level, we did similarly we did principal component analysis, and again we did see some nice separation between control and the Akkermansia treated group. The overall, I'm just gonna make an emphasis on the lipid metabolites here for this presentation. So in the feces, we did observe the significant upregulation of both primary and secondary bile acid. Primary bile acid is mainly synthesized in the liver, and then it travels to the gut and then further metabolized into secondary bile acid by the action of microbiome. And we also, so this actually, bile acid actually circulates between the liver and the gut through the enterohepatic circulation path. So it is expected that we don't see any changes in the plasma sample. So this is good. And I also want to make an emphasis on one of these endocannabinoids analog molecule called oleoethylamide, which in short known as the OEA. This was also quite upregulated in both feces and the plasma data, and which means it sort of got observed into the system. And as you can see here in this box plot, there are some significant upregulation of OEA in both fecal and plasma sample. CDCA here is one of the primary bile acid that I'm going to talk more in detail later in the slide, but I just want to make this. I just want to make an emphasis that we did see some significance in the fecal samples. And this is the result from the single-cell RNA-seq data. We were able to define 11 different cell types. And for neurons, we also did some subclustering analysis and defined 11 different subtypes of neurons. And each of these clusters were marked by distinct expression of the neuropeptides. And we also did differentially expressed gene analysis, also known as DG. And and neuron had like a highest number of highest number of DEGs, especially under the fructose condition, which is the pink color here. And we also did a pathway enrichment, and these are some of the selected pathways that we found to be interested. And interestingly, these pathways were kind of differentially regulated with different diets. So in fructose, as you can see, most of the, I mean, all the pathways are actually upregulated, while in water, most of the pathways were kind of downregulated, except for these two, thermogenesis and oxytocin signaling pathway. I mean, thermogenesis and oxidative phosphorylation pathway, which is related to energy synthesis and energy expenditure related pathway. And in the previous study, it is known that Akkermansia actually have some effect on the thermogenesis and energy synthesis. So that is somewhat consistent with the previous knowledge there. But in this study, we particularly focused on these two pathways, vasopressin regulated water reabsorption and the oxytocin signaling pathway, because these two hormones, vasopressin and oxytocin, are known to have some anti-obesity effects when it's released from the hypothalamus. And it is also important to note that we did have some large chunk of clusters in the neuronal subtypes here. And having all this omics data analyzed, and we next asked whether we can merge all these different omics together using the Mergeomics tool, which is developed by our lab. And as for the sake of time, I'm not gonna go too deep into the method, but I just want to emphasize that I use the key driver analysis in the Mergeomics pipeline to infer to find the key driver gene, which is a highly connected node in the network. So long story short, this is the network that was generated through the WKDA analysis in Mergeomics. So obviously the Akkermansia came up as a key driver. And the red and blue color here represent positive and negative correlation respectively. And we see that we again see that some of the beneficiary bacteria was upregulated, while potentially a pathogenic was negatively correlated with Akkermansia. We also see both primary and bile acid were highly connected with Akkermansia in the network. Most of them were upregulated. So this one here, phenyldeoxycholate, is the CDCA we saw in the metabolomics section. And we also found the OEA, one of the endocannabinoid analog, connected to the Akkermansia here. So having this as a result, we were able to make some conclusions. So Akkermansia, so treatment of Akkermansia seems to promote the increase of some of the beneficiary bacteria listed here, which in turn changes the total bile acid pool, which can then binds to this NAP-PLD enzyme, which is the enzyme that produces OEA. So bile acid is known to be an agonist for this particular enzyme, specifically the CDCA that we saw. So when CDCA binds to NAP-PLD, a high number of OEA is produced, which in turn binds to these two receptors, either GPR119 or PPAR alpha, which then excites the vagus nerve and send the signals to hypothalamus. and as a result, we get over expression and release of these two hormones, which then exerts the anti-obesity effect. And we did confirm the activity of PPAR, activity of these three genes here using the qPCR. So PPAR alpha obviously was highly upregulated with Akkermansia, as well as GPR119 and NAP-PLD, which all indicates the OEA is in action. OEA is actually in action. We also did some validation experiment to confirm whether OEA can recapitulate the Akkermansia effect. So we did extra experiment here, which is total of 8 weeks. We oral gavaged the OEA to the mice, and again we did see that similar patterns that we saw in the Akkermansia treatment. In the control group, there is a significant body weight gain with the high fructose diet, but here with the treatment of OEA, the body weight gain sort of like dropped. And we also checked the regulation of the expression of these two enzymes. These two genes again, PPAR alpha and GPR119. Although there wasn't a statistical significance, there was some increasing trend in PPAR alpha and both PPAR alpha and GPR119. In the hypothalamus, we did see some increasing trend in oxytocin and significant increase in ABP. And because oxytocin, we didn't get a significant increase in oxytocin, we looked further down into the oxytocin signaling pathways and checked whether the downstream genes were significantly increased. And we indeed see that upregulation of downstream genes in the oxytocin signaling pathway, which indirectly tells us that the oxytocin is indeed in action upon the OEA treatment. So in conclusion, so this is the very first study that actually used a systems biology approach to find out the hidden mechanism of anti-obesity effect of Akkermansia, specifically under the fructose diet. It seems that Akkermansia shapes the favorable gut environment for the growth of other beneficiary bacteria, while inhibiting the growth of potentially a pathogenic bacteria. And this new structure of a microbiome community seems to maintain a good balance in both primary and secondary bile acid, which then modulate the OEA biosynthesis. And lastly, OEA kind of works as a mediator between gut-brain axis, and then it sends the signals to hypothalamus, which then releases the oxytocin and vasopressin. And before I finish, I wanna acknowledge all the people who are involved in this project. I also wanna make a special thanks to Dr. Xia Yang, who always supports me and motivates me. And big shout out to all the lab members. And if you're interested in more detail, there is a preprint available with this QR code. And with that, I guess I'm open to any questions.